Hello, my name is Liz Highleyman from HIVandHepatitis.com, and we're here at the Conference on Retroviruses and Opportunistic Infections in Seattle, where we've heard some research today about hepatitis C, and we'll be hearing more presentations a little bit later in the day. So I have with me here some hepatitis C experts. To my left, we have Gregory Dore from the University of New South Wales in Sydney. And then we have Cindy Zahnd from the University of Bern in Switzerland, Kenneth Sherman from the University of Cincinnati, David Wyles from the University of California at San Diego, and Susanna Naggie from Duke Clinical Research Institute. And I'd like to start by asking each of you to describe your research briefly, starting maybe with you, Susanna.
Sure. So this morning in the hepatitis C session, I reported on behalf of my co-investigators on the ION-4 trial, which assessed the safety and efficacy of letetbasvir and sofosbuvir, which is a fixed-dose combination tablet, for 12 weeks in patients with HIV.
And that's what a lot of our audience might know as Harvoni. It's the brand name.
Also known as Harvoni. Yes. So we enrolled a large population of HIV patients, in fact 335 patients. That were predominantly genotype 1 infection. 327 were genotype 1 chronically infected. We had 8 genotype 4 patients. I think we ultimately had very good representation of African American patients at 34% and patients with cirrhosis at 20%. Ultimately, the intention to treat analysis showed an SVR, sustained virologic response of 96% overall, and this was true for genotype 1 patients. All the genotype 4 patients were cured. We found very high cure rates across multiple groups, including treatment-experienced patients and, in particular, treatment-experienced cirrhotic patients.
Excellent, thank you. David?
Yes, so in the same session, I presented data from the Ally-2 study, which also looked at sofosbuvir, now in combination with a different NS5A antagonist, the cladisvir. Also done, obviously, in co-infected patients, and we looked at both treatment-naive and treatment-experienced patients. The treatment-naive patients were randomized in a 2-to-1 fashion to either doing 12 weeks of that combination therapy or trying to see if scaling down to 8 weeks of therapy might be successful in a co-infected population. And then the treatment-experienced group had also about 50 subjects and looked at just 12 weeks of that combination therapy. The HIV-positive population that was eligible perhaps was a little more expanded in that they were allowed to be on essentially almost any antiretroviral regimen. The only exclusion specifically for an antiretroviral regimen would be a regimen that combined a mixed CYP inducer and inhibitor, so somebody that was on, say, efavirenz or nevirapine with a boosted PI. But otherwise, pretty broad inclusion criteria for antiretroviral therapy regimens. The kind of top-line results in terms of SVR12 data, we saw 96% SVR12 in treatment-naive patients given 12 weeks of therapy for genotype 1. For treatment-experienced patients, it was 98% with 12 weeks of therapy. For genotype 1, that dropped to 76% when we went down to 8 weeks. And then I failed to mention, but this study also allowed any HCV genotype. In practical terms, it really was the only other genotypes included were 2s and 3s and very few 4s. And if you added those in, really the primary efficacy numbers really didn't change substantially with those different treatment durations.
Great, thank you. Dr. Sherman?
So in addition to being an investigator on both of the trials you just heard about, my group has several pieces of work that are being presented here. 2 of them involving hepatitis C. One of them is a viral dynamic modeling study looking at one of the older treatments, the use of telaprevir with peginterferon and ribavirin in patients with hemophilia. Hemophilics historically have been thought to be somewhat poorer responders to therapy because of having mixed populations of virus. We did a small study looking at intensive sampling with modeling and were able to show that with the newer agents, including telaprevir, we seem to overcome that effect. In addition, we looked at deep sequencing of samples from one of the prior ACTG studies, also using peginterferon and ribavirin, and did show that there appears to be some selection pressure as virus is selected, even with peginterferon and ribavirin, that enhances the proportion of patients that that have dominant mutations that would be resistant to protease inhibitors. And we didn't think such selection would occur with peg interferon and ribavirin.
Okay, thank you. And Cindy, you talked a bit about some of the consequences of delaying therapy.
Yeah, exactly. The project I presented this morning is actually a modeling study where we tried to assess the impact of deferring HCV therapy until advanced liver disease versus treating people sooner, either when they have limited fibrosis or regardless of their liver disease stage, just treating them soon after their HCV diagnosis. I just want to mention that the rationale for this study was that, well, we all heard about this exciting news, the revolution. People talk about the revolution in HCV cure, but the very high price of these compounds mean that there is still a major barrier in access to this treatment. So even in rich countries like in Switzerland and other places in Europe that have universal healthcare system, the government had to impose restriction on the use of this compound and to limit their reimbursement to patients with advanced liver disease. So basically, we modeled the cohort of HCV co-infected people from the time of HCV infection throughout the different stages of hepatitis C disease and care. We assumed that people were diagnosed by being screened yearly, that all the diagnosed people will be put onto treatment with an efficacy of 90%. And we also made an important assumption on the impact of HCV therapy. So we assumed that HCV treatment would greatly reduce the risk of liver disease progression, but that it would not eliminate it completely because people who live with HIV have many other risk factors for liver disease progression. And basically what our model shows is that if people are treated soon after their HCV diagnosis, then the percentage of people who experience liver-related event is low. Less than 3% died of liver-related complication. But that if treatment was deferred, so for example, deferring treatment until liver disease stage F3 on the Metavirus scale instead of providing it in Metavirus stage F2, basically doubled the percentage of people who died of liver-related complications. It's an increase from 5 to 10%.
Okay, and Dr. Dore is going to talk about another benefit potentially of expanded treatment.
Yeah, I think the symposium this afternoon, my presentations on hepatitis C treatment and prevention, I think the results that Susanna and David presented showing the incredibly high cure rates are consistent with at least looking at the concept of whether we can use a therapy intervention to enhance treatment potential, prevention potential. Some modelling work we've been involved with shows that, for example, among people who inject drugs in Australia, if you treated 8% of active injectors per year, you would eliminate the virus by 2027. So in a modelling sense, it looks feasible. Now to take the model to implementation is a huge task. So in my presentation I'll be talking about the challenges that we face in terms of treatment and prevention. Most countries have a minority of people that are actually diagnosed with hepatitis C. There's very poor infrastructure for delivery of therapy to marginalised groups, in particular people who inject drugs, in terms of getting access to those people that may be transmitting the virus. But one of the projects that we've actually started is a project in the prison system, where we've started a project in 2 maximum security prisons, where we're having a 12-month lead-in phase, where we're looking at the incidence of new infections in the prison setting, characterising that, and then we'll rapidly scale up with an interferon-free regimen, probably from early 2016, and then look at the impact of that rapid scale-up on transmission of hepatitis C in the prison setting. So it's very exciting that we can sort of at least evaluate this as a concept. And in the setting of co-infections, another potential area. For example, in Australia, there's only about 2,500 to 3,000 people with HIV, HepC co-infection, who are largely men who have sex with men, who are generally diagnosed, generally in care at the primary care level and receiving antiretroviral therapy. So our project is to upskill the HIV GP docs who are prescribing antiretroviral therapy to take on HepC therapy treat their co-infected patients, again with a rapid scale-up where we get access to interferon-free therapy and look at the impact of that on transmission.
That's impressive, because with HIV it took a decade to start thinking about treatment as prevention, and here we are already.
Cure HepC, yeah. A little different.
So the morning session was entitled Hepatitis C, the mission is accomplished, and I think I would like everybody kind of give their thoughts about, is the mission accomplished, and if not, what might be needed to move us forward?
So I guess it depends on what the mission is. But if it's hepatitis C cure, then I think as a whole, it is accomplished. I mean, we have shown now high cure rates, over 90%, not just in HIV positive patients, which is clearly relevant for the CROI meeting, but in patients who have undergone liver transplant, in patients who have suffered decompensation of liver disease, and kind of the most difficult and neediest treatment populations. So in some way, you could argue the answer is yes. But I would argue there's always work to be done, right? So as far as HIV goes, the HIVand trial is the first phase 3 trial of current FDA-approved regimens, which means we still have a lot to understand regarding the support of safety and efficacy in large numbers and large populations of HIV-infected patients. And one of those phase 3s will never come, because I don't think one will be done with simiprevir and sofosbuvir. And we're awaiting those data for the AbbVie regimen. I think the Ally 2 trial presented today is very important, and we're awaiting the approval of that drug. While approved outside the US, has not been approved yet in the US, so it's not something that we can actually clinically apply right now. But ultimately, I guess, I think Dr. Ward said, is it with a question mark? I wish it was with an exclamation mark, but maybe it's just with a period.
Oh, my. I mean, I think that's right, and I think, as Susanna just alluded to, that John Ward kind of took people to task a little bit for the title session, appropriate so, and the big things that remain to be done, obviously, are the things Greg's working on in Australia and people need to work on all around the world, are getting these tougher to treat populations that are at high risk for both having prevalent HCV infection, but as well as perpetuating the epidemic and incident infection, getting them into care and figuring out optimal ways to deliver care to tough to treat populations. And that's a difficult thing to do.
So, I've been part of this field since the beginning, and it's incredible how far we've come, and particularly the changes in the last 4 years. But that said, I would argue that in many ways the mission now is just starting, because we have the tools to do it. We have 50% or more of people in the United States that don't yet know that they have hepatitis C. We have very poor linkage to care for those that have it. We still need to be able to assess patients in terms of their severity of liver fibrosis, and many of the providers don't even know what liver fibrosis is, and that will affect the regimen and the duration of many of the regimens we have now, and for at least the near future. We have very high cost drugs that are a barrier. We have state laws and insurers that are doing everything they can to keep us from getting those drugs and using them in our patients. So the battlefield has shifted, but the war goes on.
How about your thoughts, Cindy?
Yeah, I would kind of agree with what you said. I mean, I think it's really exciting, and yeah, mission accomplished, but as long as we get diagnose and then have an affordable treatment to provide to the people.
I must agree with Ken. I think we're just getting started. I think the affordable aspect of therapy is key. At the moment, even to contemplate hepatitis C treatment prevention as a concept at the pricing levels now is crazy.
Just
it's
to
not going to happen.
Give an idea, there's an estimated around 3 million people in the U.S. with hepatitis C, and the drugs cost approximately $80,000 and upwards. And upwards. So,
and even though Cindy's work clearly shows that treating people early is advantageous, we all have many patients with advanced liver disease that we want treatment for now, and that's our priority right now, to treat, to reduce the advanced liver disease burden. Hopefully there's enough competition within the pharmaceutical industry space that the price curve will come down over the next decade, and then we can broaden access and then hopefully if we demonstrate the treatment prevention can work, it'll be a time when we can really start to get access to people across the disease spectrum, people who are very marginalised, and to get the systems in place to really reach everyone with hepatitis C. But there's a long way to go. There'll be no landings on aircraft carriers in the next
And finally, to wrap up, does anybody have anything, sort of a message from the conference that they think is a message about hepatitis C that they think would be important to kind of make sure that it gets said?
That we need affordable therapy.
Yes.
I mean, access is key.
Access, right. Whatever that means.
Yeah, those cure rates are only so good as the people that have access to the drug.
Right.
Right,
And
and
that
you were talking
is the
about
critical
how many
focus.
people are screened. What's the screening recommendation now is at least everybody who was born between 1945 and 1965, and so far we're not doing very well with that, I guess.
But I think there's an amazing potential for empowerment with these new therapies, because at the individual level, I mean, patients feel so much better even on treatment often. And just being a healthcare professional, being able to cure almost everyone you treat is incredible.
Of course you want to do it as much as possible.
Sort of area of clinical medicine to be involved in. So I just hope that there will be this empowerment that will really drive the sector forward to a point where we can get broadened access.
Okay, well, thank you all for joining us.
Thank you.
And goodbye from Seattle.